Uh, like my friend Andy said, that tilapia culture is getting hard. And from our webinars point of view, this hardness comes from, we all know that tilapia farming is gradually switching from semi-intensive, low density and low inputs to more sophisticated, intensive farming, energy demand, and with very high inputs. This would lead to the fish getting under more stress and disease, and in turn, um, the intervention and should be a necessity by the stakeholders, by the farmers, by the decision makers to face or to meet this challenge, these stressful conditions. And feed additives, um, which are sometimes called or known as medicated feeds or functional feeds, these additives have become an important input in tilapia culture and, of course, in other species culture. So this is my talk today. And my presentation will be divided into two parts. Briefly, I will define and overview what feed additives mean, what type of feed additives, and then the second part, which is an overview of my own work and the papers which I published on this, and this has been requested by Lachance. They asked me to focus on the work we have done here in Egypt or in some other places. So feed additives, or like I said, functional substances that added to the feed, generally in small amounts, to target specific function. So we cannot just add any additive for any function. No, it should be very targeted. And that's why many additives are in use today under different commercial names. And if you look in the literature, you will find piles of publications on this issue. And the results are very varying, sometimes contradictory. Sometimes we found negative effects on the same probiotic or the same additive, and sometimes we find positive effect. Sometimes we don't find any effect. So we should be careful about which type is used and where it is used and for what species, for what size, etc. Generally, the feed additives are divided into these 5 divisions: technological, sensory, nutritional, zootechnical, and coccidostats. But in many places and many researchers just divided into 2 divisions: nutritional and zootechnical, or nutritional and non-nutritional. So you can follow whatever scheme you want. But generally, the technological additives means that we use just the technology without interfering with the nutritional aspect itself. So the technological additives influence the aspects like preservatives, binders, emulsifiers, stabilizers, acidity regulations, antioxidant, etc. So we use the technology to improve something without interfering with the nutritional status itself. The sensory division, from the word sense, which means that we improve the sensibility of the fish towards the feed, like improving the palatability, stimulating the fish through the colors, the flavors. So these are known as sensory additives. And then we come to the nutritional division or nutritional part, which means that we add some nutritional components. In this case, we affect the nutritional value of the feed. So we add these specific nutrients required by the animal, like vitamins, amino acids, trace minerals, etc. So these are parts of the nutritional requirement of the fish. That's why they are known as nutritional. Then we come to zootechnical, which are not nutritional, but they improve the nutrient status in the animal by improving the utilization of the fish, of the feed in the fish, improving the flora in the guts, the beneficial microbes in the gut, so that to improve the digestibility and absorption and the health of the gut and of the fish overall. So this is known as zootechnical additives. And finally, the coccidostats and histomonostats, which are generally used in poultry and in other land animals to control parasites in the guts, to control gut parasites. So these are the quickly the divisions or the classifications of feed additives. In the market, you can find these common additives under these different the 5 different additives. You can find that probiotics, prebiotics, synbiotics, postbiotics, enzymes, plant-based, which is phytogenics, organic acids and organic salts, immunostimulants, antioxidants, phospholipids, vitamins, binders, etc., etc. Many types under different commercial names you can find. Okay, now, what are the functions? Like I said, each additive should have a specific function or specific functions, but overall, they can lead to what you see now in the screen, promote the growth, disease resistance, and of course, enhancing the immunity and antioxidant, prevention of oxidation, like they are antioxidant, like I said, antitoxins, improving feed properties, like I said, improving feed digestion, like enzyme, for example, improving product properties and quality, etc., etc. Oops. But like I said in the beginning, sometimes you find negative effect. We use the same additive, but we can get negative effect, or even, like I said, non-effect of feed additive. So we should be careful, very careful. Feed additives may not be effective. They may even, like I said, pose negative impacts. They may be effective in one species, but not in the other. They may be effective in one farming system, but not in another farming system. They may be effective at certain doses, like we will see in just a few minutes. At certain doses, the additive were very effective, but when the dose increases, negative effects or negative impacts occurred. So we should be very careful about that. Therefore, before applying feed additives, farmers should be very careful about which species and which size, because sometimes they can be effective in the like larval stage, but not in the grow out, or vice versa. What type of feed composition? What are the quality or the qualities of the components of the feed? Because the more the feed components are in good quality, the less we require for the additives. Which farming system are you using? Is it a RAS system, a recirculating system, or semi-intensive? In semi-intensive, the fish can get access to live food in the pond, so they can and the live food can provide some of these additives. But if you are working in a RAS system, for example, most likely you will need these additives. Also, like I said, the dose of the additives. This slide is very important. We should be very careful. Okay, so quickly, what are the probiotics? There are different definitions for probiotics, but generally they are products, beneficial products produced by microorganisms like bacteria, yeast, etc., which stimulates the growth of another microorganism. So we add this product to increase the microorganism, which in turn improves the welfare of the animal, target animal, and combat or fight the pathogenic microorganisms. This is one definition. The other definition is, which is accepted by the FDA and WHO. Simply, they are like live microorganisms. When they are administered in adequate amounts, they can confer or produce or provide health status to the host animal. Why probiotics? Probiotics, from the definition, they improve the status of the fish. Previously, most of the farmers, either for the fish or the large animals or poultry, they were using antibiotics. And as we all know now, there is a increasing concern about the risk of using antibiotics, especially the human health risk and environmental risk. So there is a public awareness about reducing or avoiding antibiotics, and that's why probiotics have become a very important and ideal alternative for antibiotics. And the functions of the probiotics, especially in fish, like I mentioned before, growth and immunity, stimulation and antioxidation, and inhibition of pathogenic bacteria, and of course some other functions depending on which probiotic we add. How probiotics work to improve or to confer beneficial effects. This is done through production of inhibitory substances, like, for example, hydrogen peroxide, bactericides, etc. And these products block the adhesion site of pathogenic bacteria. And we will see in a few minutes one really nice slide we have published indicating that when we add these probiotics, they block the adhesion sites of the pathogenic bacteria and increase the adhesion of beneficial bacteria to improve the gut criteria and gut health, and in turn improve the immune stimulation and digestibility and absorption, etc. They can also degrade the toxins through blocking toxin receptors, and also they can modulate immune responses through the expression of specific immune genes, okay, like we will see in a few minutes. They can maintain the balance of intestinal microbes. There, in each animal, there are beneficial microbes, beneficial bacteria, and pathogenic bacteria. They are living in a balance. Under stressful conditions, the pathogenic bacterial number increases and the beneficial decreases. But when we add these beneficial probiotics, they improve or increase the number of beneficial microbes and reduce the number of pathogenic bacteria, and therefore they improve the gut health and the overall health of the fish and reduce mortality, improve survival, etc. They can also increase the fish's resistance, of course, like I mentioned, by reducing the pathogenic bacterial number. They can improve the appetite if we add a probiotic that is related to appetite and stimulate sensory receptors in the animal. And finally, they will enhance nutrient digestion and absorption. Yeah, this is the photo I'm talking about. In tilapia farming, the most common probiotic is lactobacillus bacteria. This is very, very common, and hundreds of papers have been published on that. In this photo, we extracted marine lactobacillus from the bottom and the water, marine water, and then we produce it in mass production. I will talk about that later on. But in this photo, you will find the one in the left, the lactobacillus is adhering to the wall of the intestine, while the one on the right, the control diet, which did not have these probiotics, they were almost free of any adhesion of the bacteria, of the beneficial bacteria. This was a scanning microscope photo. But the one on the left is not mine. This is I got it from the net. This is sorry, Streptococcus bacteria. So this is what should be said about these common beneficial bacteria, probiotic bacteria: Lactobacillus, Enterococcus, Streptococcus, and Bidococcus. This is what I've just said in the previous slide, that through this scan, it's just a description of the photo I talked about, which meaning that the adhesion of the beneficial bacteria to the wall of the intestine, which improves the performance of the fish. Then we come to prebiotics. Prebiotics are generally not live animals, or not live bacteria, not live organisms, but they are products, mainly carbohydrate products. They are non-digestible fibers, which is non-digestible carbohydrates, beneficially affect the host health through stimulating the growth and activity of other microorganisms. So we add the prebiotic to improve the growth of the probiotic. Okay, so it's like a two-step effect. We add the prebiotic, we improve the lactobacillus, for example, which improves the animals, the fish's performance and immunity and health. So this is like I said, substrates. They are used as substrate that is selectively utilized by the host microorganism, which conferring the health benefit to the fish. Since they are carbohydrates, so they are mostly either oligosaccharides, which is we have many types of saccharides, starch, lactose, lactosucrose, chitosan, pectin, and many other carbohydrate fibers or non-digestible carbohydrates. So these are the prebiotics. Then the synbiotics are a combination of both. So we add prebiotic, like I said, and probiotic, so they both help each other, so they work synergistically, improving the effect of the fish. Then we come to what we call postbiotics, which contain, it's like a probiotics, but we used inactivated microorganisms. So we treat the organism, the bacteria or the yeast or whatever the microorganism is, the beneficial microorganism, we treat it like heat treated, for example. And then we take their metabolites and use it as an additive in the fish. So they can deliver different benefits to the fish without adding or without any risk linked to the live bacteria, because sometimes the live bacteria may impose some risks or some negative effects. So using postbiotics avoid this effect since they are heat treated, almost dead. Then they can enhance the beneficial microbes, offering a safe and effective microbiome supporting strategy. So this was just like an introduction of the additives generally. Then we come to briefly some of our work. We carried out several studies on the use of feed additives, and these are the types we covered. We covered different probiotics and prebiotics. We used enzymes. We used plant-based blends. We used emulsifiers. We used organic acids. We used sensory additives and many others. And in the end of my talk, I summarized these studies and provided the source so that you can get the papers quite easily. So I will go quickly over these. This is the system, or these are the systems we used in most of these studies. The one in the right-hand side is a closed system in my lab. This tank is 250 liters. These are 12 tanks. And in the bottom, there are the rest of the system. And also some of the studies, we ran the study in the lab and then confirm it in the field. So we use one of the farms in Egypt here, and we use these happas. This happa is 2.5 meters, cubic meters, but the water capacity is 2 meters. So we most of the studies were in these on these systems. This is just some of the snapshots of the titles of some of these studies. And almost all of them were published in top reviewed journals. The first one, using the emulsifier, a commercial emulsifier. And in these slides, I focused on the health part of the study, not on the growth, because most of these studies resulted in a gross performance, improving gross performance and feed utilization, digestibility, blah, 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 blah. But I was asked to focus on the health and immunity of the fish. So I focused on that. So we added different levels of this emulsifier, which was called Lyso-max and maxillase. And we find the curvilinear, immunity parameters and the digestive enzymes on the left were improved with increasing the level at certain level, and then was reduced later on. When we ran the quadratic regression analysis, we found that about between 4 to 6, between 4 to 45 gram per kilogram, gram per kilogram, yes, was required for best digestibility or digestive enzyme. Same thing for the immunity parameters, lysozyme, superoxide dismutase, ACH50, etc. So the same curves were obtained. This means that this product, about 0.45 gram per kilogram, was effective for the fish. And in fact, immediately after running this trial, I recommended this product, and it is being used by a number of factories, which I'm now consulting. And this is a digestive histology in the anterior gut, mid gut, and posterior gut. We found, it's not very clear here now, but we found that the goblet cell number and the villi, intestinal villi, were all increased with increasing the level of this emulsifier up to certain level, and then reduced. In another study, in my lab, we published a number of papers on isolating marine bacteria from marine, and then we mass produced these bacteria and use it as probiotics. It was, and we published like about 5-6 papers on this track. So one of them was the marine lactobacillus. We extracted this lactobacillus from the mud, the sea bottom, and then we mass produced it, and then we add it in the diet into different levels, 0.5 and 1%, and then after that the fish was challenged with Eromonas hydrophila. At 1% inclusion level resulted in the best immune response, the best cytokine gene expression, interleukin 4 and 12, and IFN genome gamma, and then the gut health after the challenge. This indicates that appropriate level of marine lactobacillus, which is 1%, can effectively enhance tilapia resistance against pathogenic bacteria. And the graph on the left, after challenging the mortality, and then we will see that at 1%, the mortality was the lowest, while the control was the highest. And the graph are saying the same thing. We'll find that the immunity increased and then decreased later on. And the same thing for the genes. I'm afraid the time is running. Again, we isolated what we called secrophile bacteria. It's a cold-resistant bacteria. So we isolate this bacteria also from the sea, and then included it into the diets at different levels, 0.5 and 1%, and the results were the best at 0.5% of these Secrobacter bacteria. And in fact, it's called Secrobacter bacteria SO80, which is SO is initials of my colleague's name. That was part of her PhD. So she named the bacteria after her name. Yes. You can see the details later on. Then we can come to the botanical blend, the phytogenic bacteria, phytogenic products. This is a commercial product from France. It's called Phytoaquanite. And again, we added different levels, 250 grams, 500 grams, 750 grams per ton. And then we also, and this was in the field, not in the lab. It's a field trial. And the optimum response was at about 7 to 8 gram per kilogram, depending on which parameter we are measuring. But this was using the quadratic regression. And like you see in the graphs, it indicates that the performance improves at certain level and then deteriorates. The one on the left, on the right-hand side, is the beneficial bacteria and the pathogenic bacteria. Adding these plant blends improved the beneficial bacteria up to 0.75 and then deteriorates, while the broken lines, which is pathogenic bacteria, was decreased with increasing the level of this phytogenic product, meaning that this product improved the gut health of the fish. Then we come to Phytase. We ran two trials, one in the lab, adding different doses 1,000, 2,000, 3,000, and 4,000 FYT per kilogram. We found that the best growth, immunity, and antioxidant parameters were achieved at 2,000 unit using this quadratic regression. That was the lab trial. You know all that phytase is used mainly to release phosphorus from the products, because phosphorus is chelated with phytic acid. So phytase breaks this link and liberate the phosphorus. That's why we added it. So in the lab trial, yeah, this is the same lab trial. The liver function enzymes were reduced with increasing the level and then increased after certain level. In the lab trial, we took the level produced the best performance, which is 2,000 units, and we had three trials or three treatments. One, we added 10 kilogram monocalcium phosphate without any phytase, which is commonly used in commercial feed in Egypt. This is the amount added to the commercial feed. And then we added, in the other trial, we added the phytase without the phosphorus. And in the third trial, we added 50% of both, 5 gram per kilogram monocalcium phosphate plus 1,000 FYT per kilogram. The results, as you see in the graph, the field trial indicated the complete replacement, this is the good thing about the trial. We successfully replaced the phosphorus completely and added just the enzyme. And there was no need for adding the phosphorus in terms of monocalcium phosphate or dicalcium phosphate. Not only that, but the profitability increased by 10.5%. And immediately after finishing this trial, I also recommended this enzyme to be used commercially, and a number of feed producers in Egypt are currently using this enzyme as an additive without adding phosphorus. And this point is crucially important, because in this case we improved or we increased the profitability and we reduced the environmental impact, because the phosphorus we add, most of it will end up in the environment as discharge in the environment. So we eliminated this point. So this study, I believe, was one of the good studies we carried out. For the sensory studies or the sensory effects, we studied the effect of different colors, and this was a master degree for one of my students. We added different colors: red, green, blue, yellow, and control, which was a brown color, in the diets, and fed to the larvae and the fingerlings. The larvae were affected, and the dark colors, which dark blue and dark green and dark brown, produced the best performance, because the contrast between the feed color and the bottom of the tank, these contrasts were very clear. So because we know that the larvae are visual feeders, they use the vision to feed, but the fingerlings use the other sense as stimulators. So they were not affected. Larvae affected, fingerlings were not affected. So this study also was a nice one. Was straightforward, but the results were convincing. And this was published in Aquaculture Nutrition about 10 years ago. Finally, I'm sure I took a lot of time for your time. I'm sorry for that. This is like a summary of the effect of different feed additives on growth, feed efficiency, blood parameters, immune responses, antioxidant parameters, and overall health. So we used the name of the additive, the dose we used, and what effect they had. and then the authors, and I put the DOI. So you can just take this DOI, copy it, and then you can quickly get the whole paper. So I don't want to go through all these in details. You can easily track this. I think that's it. And I always end my talk with my book. This is my book, Tilapia Culture. The first one was published 2006. The other version was just 2020. Thank you very, very, very much. And I'm sorry for taking more time than I should have taken. Thank you very much.
Okay, thank you, Professor Said, for your valuable insights. Thank you very much. And as we all know, Professor Said shared the insights from international perspective, especially on the practical use of feed additive in tilapia farming. So now it's time for Q&A session. If you have any question about functional feed additives, alternative use solution, please feel free to ask. I see there already have many questions for you, Professor. They are very interested for your topic.
Well, tell you something. If some of them are from Egypt, they can contact me because, yeah, there are about 10 questions here or probably more. It would be a little difficult to, but the first one, if you have any question, sorry. Hello, Prof. To what extent do streptococcus, no, this is not for me. Dr. El Sayed, many thanks for your, this is, thank you very much. That's my pleasure. Mohammed El Naqib. Greeting, thank you, Andy. Greeting from Egypt. I have high respect for the valuable information you shared. Thank you very much. So it seems the impact is difficult to quantify. However, this is the answer of Andy, I believe.
There are some questions. May I read for you? The first question, what the more effective binder to deliver the functional additive to tilapia in the diet?
What can you say it again, please? Which is more
effective binder? Can you see in the
effective binder?
Yeah.
I tell you, I'm one of those who use additives at very, very limited scale. In most of my work, I don't recommend additives, just one or two additives at all. If you have extruded feed, okay, if you have an extruded feed, you do not have to add any additive because the extruded feed itself, the extrusion gelatinize the feed and make it, very good, compacted and durable. So you don't have to add any additives. If you add like compressed feed, I think that carboxymethyl cellulose is a functional one, but you add it at probably 1 or 2%, not more than that. Sometimes they add starch as an additive, as a binder, and as a source of carbohydrate. But this also depends on the cost and the type of feed you are producing, extruded or pelleted or mixed. What do you call it? Pelleted feed. So yeah, different one. But like I said, CMC is the one. Or no, you don't use anything if you have extruded diets.
Thanks, Professor. And
also, sometimes they use, alginate agar, but these are very expensive. So I don't recommend at all. Okay.
And also there another question. Professor, how can we measure and keep effective live probiotics in feed during processing, storage, and feeding?
Live probiotics? No,
Yeah.
we don't add live probiotics. We produce the probiotics in the lab, okay, and then
The viable probiotics, maybe.
we process them to become like a powder, like a product, okay. So you have treated probiotics, but we do not add live because processing will kill them anyway. So that's the way they are included. Even commercial probiotics, they are not live. They are either dead lactobacillus or metabolites of these bacteria, or even yeast. You can use the yeast as yeast, but dry yeast, okay, and then include it to the fish. So they are not live probiotics. You may add live probiotics to the water if you want to improve the water quality. I was in Ecuador a few years ago, and the farmer was growing a huge amount of lactobacillus in his tanks and used to get these lactobacillus live and grow them in the pond. So they, get reproduced, and they get, the number increased to improve the water quality. So in this case, yes, you can use live probiotics, but in the feed, most likely not.
Okay, thanks, Professor. Also, another question about the standardized probiotic levels using CFU per gram. Maybe the dosage.
Yes, yeah. Look, this depends on which probiotic you are using. If you are using enzyme, for example, this is different units. But mostly for the bacteria, we use the count, the number of bacteria per milli or per, and then we use that. We transfer it into grams, okay, and then we add it as percentage in the diet. But mainly it's based on the counted bacteria. But for, like I said, for plant trial, we used weight. This is gram per kilogram. For enzymes, also this is unit. For organic acids or organic salt, we use different dose, different weights. Only for bacteria or for live organisms, we use the counting.
Thanks, Professor. Also, many questions. What are the effective of high-level functional additives on fish health? Are there any negative effect? High-level functional additive.
No, I didn't understand the question really. So
maybe
can you repeat it?
Maybe the high-level functional additive will do some negative effect for the
yes,
fish health.
Yeah. And if you look at my work, you find that after a certain level of the inclusion level, the effect will decrease. Like the immune response increase with lactobacillus up to 1%. And then after that, it was reduced. And I said this in the beginning of my talk, that sometimes they can get negative effect if the dose is higher than what's required. So we should be careful about the dose we include, because they can work, instead of working synergistically, they can work what we call antagonistically. Antagonistically means they can add negative effect. So you are right. Higher level beyond the optimum level may pose negative effect.
Okay, thanks, Professor. And single probiotics more stable than mixed probiotics in fish feed?
A single probiotics? What?
Yes, and the mixed probiotics more stable, which is stable one. Single strain probiotics.
Sometimes, like I said, there are sorry, tens of probiotics, hundreds of types. So it depends on which type you are using. The enzymes, for example, we cannot add the enzymes with the feed because they will be broken or they will be broken. So we add the enzymes after the feed is produced. We spread them on top of the feed. Okay, so it depends on what type of enzyme you are adding. If you add organic acid or the plant materials, we add that with the feed. So this depends on whether the probiotic is heat liable or can resist heat. If you add, like, for example, vitamin C, it can resist heat, but certainly some is getting lost. So we generally add more than the fish is requiring, a little bit, to be able to get what the fish is requiring after processing. So just to summarize this, the adding the probiotic with the feed or without the feed depends on the type of the probiotic and whether it can resist heat or not. But most of them are added with the feed anyway.
Thanks, Professor. And also another question: how can feed additives affect on farm and didn't affect on another farm?
This farm, like I said, the farmer's growing tilapia semi-intensively, for example, and he adds fertilizer, and he has a lot of phytoplankton, zooplankton, worms, and different live food. So the fish can simply get sufficient amount of whatever the fishes need, and then they can improve the health and improve the performance, etc. In another farm, like I said, the farmer is using RAS system. There is no live food in the system, so it can be affected by the additive. The water quality, if the fish is living in, for example, low temperature or high temperature, higher than what the fish is requiring, adding the additive may improve the performance and immunity of the fish. So the water quality can affect the farming system, can affect the fish size itself, like I said, because the resistance of the fish can vary from one stage to another. The source of the fish. One farmer brought the fish from bad hatchery, so the fish is weak and stressful. The other farmer get his fish from good hatchery, so the fish is in good health, and then the effect of the probiotic can be different. So yeah, there are many many factors can interfere in improving or not improving the performance of the fish when we use these additives. I'm not sure whether I answered or not, but I hope yes.
Okay, thanks, Professor Said, for your patient and detailed answers.
Thank you very much. It has been a pleasure, and once again, I'm really happy to be with you, and I hope we continue cooperating. And thank you for the audience. We still have about 60-something people, which is good after 2-3 hours still waiting with us.
Yes, thanks a lot. And also there are still many questions. We will collect it and we'll answer it in writing way. Thanks.
Okay. You can send them to
Thanks a
me, and
lot
I hope I
for
will
the
be able to.
questions and
Thank you.
answers.
Thank you, Andy, and thank you for all of you, and we'll see you again.
Okay, thank you, Professor Said. Thank you again.
Okay, see you. Bye-bye.